blood-brain barrier is formed of endothelial cells that compose a very selective semi-permeable border. The only way you're getting in really is if you're lipid soluble or if you're very small. In the bloodstream there is a constant fluctuation of concentrations of different substances including ions, glucose and hormones, as well as a presence of toxins such as bacteria. It is important that the brain is not exposed to such fluctuations and neither is exposed to these harmful toxins. The membrane of the neurons are very sensitive to fluctuations in ion concentration, so that is another reason why we need to keep the brain and the blood separate, other than to protect the brain from toxins. The iron concentrations are really important to keep that membrane potential stable. So on the right there we have two molecules behind bars. Above we have dopamine, below we have GABA. These are neurotransmitters used by the brain to transmit signals. Without the barrier there, these neurotransmitters would be flushed into the bloodstream and the neurons would lose that availability, that immediate availability. So we need them to be held within the brain so that they can perform their function as well as they do. We're now going to discuss the different types of capillaries that we find in the body to better understand the ones that work at the blood-brain barrier. There are three types of capillaries in the body. The first is known as a continuous capillary. This continuous capillary forms very tight junctions. There's no fenestrations and substances have a very difficult time making their way out of the capillary when they're surrounded by continuous endothelial cells. Next we have the endothelial cells of a fenestrated capillary. These cells are not found at the blood-brain barrier because substances would find it too easy to make their way from the brain back into the bloodstream and vice versa. So we find these at other areas of the body but not at the blood-brain barrier. And then finally we have the most easy access capillary. It does have fenestration. They also have very loose gaps within each cell and as you can see they're a lot more permeable. So how come continuous capillaries have such tight junctions? Well, if we look closer at the junctions, what you can see in this drawing is there is almost a thread that tightens the endothelial cells together, and this thread of course is a protein, and the two main proteins are known as occludin and claudin, and these proteins essentially thread together the endothelial cells of continuous capillaries. Usually there are three routes of which substances can move through endothelial cell walls of capillaries. The first is vesicular transport or transcytotic vesicular transport, and this is how proteins and large molecules will make it to the other side of the capillary, and this uses vesicles and receptors, and we will go through this in a bit. The second route is known as the paracellular route, and this is where substances will move through the gaps between the cells. So obviously this is very limited within the capillaries of the blood-brain barrier due to the tight junctions. And then finally the transcellular route, and this is where substances will move through the cells, through those holes that we just mentioned. So the tight junctions found between the endothelial cells of continuous capillaries is the main aspect of the blood-brain barrier. So it's important that the proteins that do thread together these endothelial cells are not disrupted and they do not break down. However, if they do, the astrocytes have end feet that completely surround the capillaries within the brain, and if there is a disruption, the astrocytes have the potential to produce signals that will induce the production or synthesis of the proteins occludin and claudin or other proteins that thread together the endothelial cells, and this ensures that the tight junctions are kept tight. So far we've looked at how structure of endothelial cells in continuous capillaries relates to the function of the blood-brain barrier, and this includes the tight junctions found between the endothelial cells and the lack of fenestrations within the endothelial cells. There is also very poor vesicular transport, and we're just going to have a quick look at what vesicular transport is. So here we have the inside of a capillary, continuous capillary, and there we may have a hormone in the blood, and the hormone will bind to the receptor, the membrane will pinch around the hormone, and the hormone or molecule can make it to the other side to the brain, and this is known as transcytosis. But this mechanism is very poor at the site of the blood-brain barrier. So lipid-soluble or hydrophobic molecules can pass the membrane since the membrane itself is made of lipids. But what happens if we have a lipid-soluble toxin? Well, what we have inserted into the membrane of endothelial cell are known as non-specific drug transporters, and these transporters use ATP to actively pump out any lipid-soluble toxin that's made it across the membrane. Now, it's very useful if the toxin itself is harmful. However, drug developers find this to be the hardest barrier to overcome, since drugs that will have therapeutic effects within the brain are actively selectively transported out of the brain through these transporters. So sometimes a mechanism to deactivate these transporters is looked at so that the drug can reach the brain. The most abundant non-specific drug transporter is known as p-glycoprotein. So we know that lipid-soluble or small polar molecules can pass the membrane pretty easily. However, glucose, which is a slightly large molecule, is definitely needed in the brain, but it needs a special route to reach the brain. So what we have on the endothelial walls of the blood-brain barrier is known as a glucose transporter. These glucose transporters are called GLUT1 transporters, and GLUT1 transporters are not insulin dependent, unlike other transporters within the body. We do not want the function of the brain to be dependent on levels of insulin. We just want the glucose to be able to reach the brain whenever, since it needs a high amount of energy. Another important transporter that we haven't mentioned yet is one that is for amino acids. Amino acids are important since they are the building blocks of protein, so we have specialized receptors within the endothelial cell walls of the blood-brain barrier that will trigger the transport of that amino acid to the brain. In Parkinson's disease, dopaminergic neurons in the substantia nigra disintegrate, therefore basal ganglia motor neurons are compromised, as is motor movement. Due to the disintegrated neurons, there is a lack of dopamine. However, dopamine cannot pass the blood-brain barrier, but what we can do is insert dopamine's precursor into the bloodstream. This precursor is known as L-DOPA or levodopa, and this amino acid will bind to the amino acid receptor and be transported to the brain, where it can become dopamine. If you found this video helpful, give it a like. If you want to see more, subscribe, and if you have any requests, please comment below. Thanks for watching.